So let's continue our discussion of microbial genetics. What is a mutation? Generally speaking, it's a change. A change to the genetic code. You're a mutant, I'm a mutant, everybody's a mutant in a literal sense because we've all had changes to our genetic code, particularly since we were conceived or brought into existence. This change to our genetic code is essential to life on our planet. It's the driving force of the theory of evolution via natural selection. Any change to any nucleotide sequence in any genome of any organism is considered a mutation. Most mutations we don't really notice. They occur all the time, and they will occur in cells that aren't necessarily going to be that missed. So we'll have cells in our bodies that occur as human beings that accumulate mutations, and those cells will be selectively killed off or die off very frequently. For we as human beings, generally speaking, we notice those mutations when the cells that acquire those mutations do not die off like they are supposed to, but instead become cancerous. In microbial populations, we typically notice those mutations when microbes develop new traits, e.g. antibiotic resistance. These mutations are noticeable when the genotypic change leads to a change in the phenotype, so when we can see a change occurring in the population. These mutations can involve losses of base pairs, sometimes the addition of base pairs, or the mixing and matching, the rearrangement of the base pairs. So let's first talk about the wild type. The wild type is going to be the organism that doesn't have the mutation in your gene of interest, your favorite gene. So when doing studies on an organism, you'll have a wild type that has a non-mutated gene that you compare to your mutant strain or your transgenic strain. And that mutant strain is going to have the mutated gene with the mutated characteristic. This mutated strain, generally speaking, is going to have a change in its morphology, its shape, maybe it'll change in its metabolic characteristics or nutritional characteristics, or its control mechanisms. Maybe it becomes resistant to a chemical that formerly would destroy it, or it can have a temperature preference that it may have gained or lost, or it could just have something crazy happen with an enzyme. There could be some change to the organism that we observe due to the mutation that occurs in the mutant strain. What can cause a mutation? Sometimes they're just spontaneous. Every once in a while, DNA replication goes bad, and there's a mistake that's not proofread by the DNA polymerase III, and it gets incorporated into the new chromosome, and then that mutation is passed down. Sometimes, though, the mutation can be induced. We can cause that mutation to happen by exposure to specific environmental factors. What are some of those environmental factors, or what are those things that cause mutations? Something that is known to cause a mutation is a mutagen. And those are primarily going to be either chemical or physical in nature. So let's talk about two common mutagens. We have radiation-based mutagens. Those can be ultraviolet light or X-rays. Those are kinds of electromagnetic radiation. Or we can have chemical-based mutagens, aka nitrous acid, which, strong stuff. Don't mess with nitrous acid. Nitrous acid and nitric acid scare the living daylights out of me, which is why I don't like messing with those liquids. I digress. Back to mutations. So, let's talk about a point mutation. A point mutation is when we have one specific change to a specific nucleotide. So it's very pointed. It's very focused in its target and change to the genome. It can be the addition of a specific nucleotide, an A, C, G, or T, or it could be the deletion of the specific nucleotide, or it could be a flip-flopping. So instead of adding an adenine or deleting adenine, you could take the adenine and turn it into a guanine. We also have missense mutations and nonsense mutations. And I don't mean by nonsense, I don't mean like stupid that don't make sense. I mean that it's going to be a change to a normal mutation into a stop codon. So a nonsense mutation will take and introduce a stop codon. And actually, this is a typo here. A missense mutation changes a normal codon into a stop codon. A missense mutation is going to be any other change to the genetic code that leads to a different amino acid. So missense mutations introduce different amino acids into the host, or into the protein that's being transcribed and translated by that gene. These missense mutations are going to involve a change to the codon that codes for that specific amino acid. Now, if we have an amino acid that's on the surface of the protein, and we substitute one nonpolar amino acid for another nonpolar amino acid, it's probably not going to be that big of a deal. But let's say we're working with an enzyme, and in the active site of the enzyme, we substitute a positively charged amino acid with a nonpolar, neutrally charged amino acid within the active site of the enzyme. That'd be a catastrophic mutation to the protein. So these mutations, depending on the amino acid substitution and the kind of substitution and where the amino acid is located within the protein, can cause no change, or it can cause the protein to completely stop working, or it can cause the protein to work slightly differently. So missense mutations are a change in the codon that change the amino acid. A nonsense mutation will take a codon for a regular amino acid and turn it into a stop codon. Silent mutations will alter the base but not change the amino acid. Remember that redundancy. There are more codons than there are amino acids. So multiple codons can code for the same amino acid. So if I change a codon that codes for glycine to a codon that codes for glycine, you're still going to have a glycine amino acid in the protein. Even though there's been a mutation, that mutation still ends up with the same amino acid in the protein at the correct spot. So who cares? It's a silent mutation, completely unnoticeable, and it will have zero effect to on the organism. Well, I shouldn't say zero. It'll have very minimal effect on the organism. We could also have a back mutation. This is when we could have a gene go from a mutant state back to its wild type or original base composition. Back mutations can also occur. They do not occur very frequently, but hey, it can happen. And then we also have frame shift mutations. These frame shift mutations are catastrophic mutations, generally speaking. These can occur when DNA bases are deleted or inserted into the genome. Now earlier, when we talked about transcription and translation, I mentioned how there's that start codon, that AUG for the start codon. And that AUG for the start codon was very important because it gives us the frame of reference for reading each codon downstream. And depending on your frame of reference, you could end up with very different amino acids in the protein. By changing our frame of reference with a frame shift mutation, we are going to alter essentially all the downstream amino acids in the protein and potentially introduce a stop codon where there was formerly no stop codon. Frame shift mutations in almost every single scenario I can envision result in a non-functional protein being produced. This is such a beautiful summary table. Let's spend some time talking about it. So here is our wild type gene, our normal gene. Here's our DNA triplets, TAC, TGG, CTG, so on and so forth. Then it goes to our messenger RNA, and then here's the amino acids that get in it, our methionine, threonine, aspartate, glutamate, methionine, lysine, so on and so forth. Now let's have a missense mutation. Remember, a missense mutation changes the codon so that we go from one amino acid to a different amino acid. In this case, we change this G to a T, which means the messenger RNA now has an A, and now GAA is the codon that codes for a glutamate as opposed to an aspartate amino acid. So this protein now has a new amino acid in it. It may or may not work well. We could have a base substitution that's silent, so there'll be no change in function. So instead of having a CTG, we could have a CTA. Both CTG and CTA will code for codons in the messenger RNA that both code for the same amino acid. So we had aspartate in the original, but then we mutated and introduced an adenine, but we still have an aspartate in the protein. So there's no change to the amino acid. It was a silent mutation. Now let's talk frame shift mutations. But I can delete nucleic acid. In the original sequence, we have TGG, CTG. If I delete a G, I'll have TGC. C, T, C, G. Everything downstream is shifted. And all of the downstream codons have shifted as well. So instead of having an aspartate, I have a threonine. Instead of a glutamate, I have an arginine. And instead of methionine right here, I have a stop codon. Non-functional protein. This is going to result in a protein that is dramatically different and no longer performs the required function. We could also have a frame shift mutation where we insert an extra nucleotide. So, in the original, we had a TGG, CTG. So there's two G's in a row and then a C. If I insert a third G, so TGG, GCT, I've now inserted an extra nucleotide, and all the downstream amino acids are going to be changed, or downstream codons will be changed, and downstream amino acids will then be changed as well. So we went from aspartate, glutamate, methionine, lysine, to arginine, aspartate, glutamate. So frame shift mutations, by inserting or deleting a specific nucleotide, you can have a catastrophic change to the genome. Sometimes, though, it's kind of a pain in the butt to look at this genetic code and these frame shift mutations. So let's have some fun with some Dr. Seuss-like sentences. This will help it make a lot more sense. At least this did for me when I was a student initially going through it. Assuming Microsoft Word will load. Come on, Microsoft Word. Please load. Oh, good gravy. My computer is slow. Blank document. So, let's make the font size nice and happy and large. 48 is pretty fat. The cat, well, let's say, the fat cat ate the bat. So here's a series of a sentence that is arranged with three-letter words. These three-letter words correspond to codons. Now, let's have a frame shift mutation. Oh, this shouldn't be that difficult. I just changed the orientation. There we go. So I am going to delete the letter F. The, at, atta, etha, iba, ta. So by deleting one letter, I changed all of the downstream words to be nonsensical. I changed the meaning of the information contained. This is a frame shift mutation in my sentence. We go back to the PowerPoint here. So frame shift mutations can change, just like they can be catastrophic to that terrible sentence I just had. A frame shift mutation can be catastrophic to the downstream codons and the subsequent amino acids that make it into the protein. Oh, back to full screen. Here we go. There we go. So how can we repair the mutation? Our organisms, microorganisms and eukaryotic organisms, so we have some of these as well, have DNA repair mechanisms that are constantly proofreading the genome to make sure that we fix these mutations if they come up. And we're able to catch a lot of these mutations and fix them before they become permanently incorporated into the genome, but we don't catch them all, unfortunately. We have additional systems for finding and repairing DNA that's been damaged. In addition to the DNA proofreading mechanisms, we are not going to go into tons of depth for these additional mechanisms. This is an introductory microclass, not a molecular bio class. First, let's talk about UV damage repair. Ultraviolet light causes lots of damage to cells. Ultraviolet light is going to cause two adjacent thymine molecules to be dimerized together. We can repair this damage through the use of an enzyme known as photolyase, which is wonderfully named. It lyses out the damaged sections of nucleotides that have been damaged by the photovoltaic, or the photons of ultraviolet light. Photolyase, though, only works for a very small number of UV mutations. There's just no way that photolyase can keep up with the cell being bombarded with thousands of ultraviolet, or billions of ultraviolet photons all at the same time. Cells that cannot repair very severe widespread UV damage are going to eventually become cancerous and or die. I use ultraviolet light in a laboratory setting as a method of sterilizing my goggles, for sterilizing my glassware. I can use ultraviolet light in my heat exchange system in my central air of my house to remove pathogens and mold growth from the heat exchange coils. And this same kind of light is the same kind of light that individuals expose themselves to when they go to tanning booths, which is one of the reasons why individuals that frequently go to tanning booths are at increased risk of developing skin cancer. Let's say we have a mutation occur, and this mutation wasn't necessarily from a thymine dimer occurring from ultraviolet light mutating the genome. We can still have excision repair. So this is just going to be a generic method of repair where we find a generic mutation. The enzymes are going to involved with excision repair are going to break the bonds between the sugars and phosphates in the DNA background. So they'll cleave those covalent bonds where the error mutation has occurred, and then they're going to remove the defective nucleic acid and reinsert the correct nucleic acid, and then we'll patch up the repair by forming new covalent bonds between the phosphate and sugar backbone of the DNA. DNA polymerase 1 is involved with this excision repair, and ligase is an enzyme that will catalyze the formation of a covalent bond between the phosphate and the deoxyribose sugar in the DNA backbone. So, let's say we need to chop out, here's a mutation that's occurred. We're going to cut out this mutation from the piece of DNA, and then we're going to insert the correct nucleotides and then patch them in place. So there are pros and cons to mutations. Sometimes we like them, sometimes we don't like them. Mutations that are permanent and inheritable will be passed from parents to children. They'll go on to the offspring from generation to generation, and they can also go from virus to virus. Most spontaneous mutations are not beneficial. I want to strongly emphasize that the overwhelming majority of spontaneous mutations are not beneficial. There's a very, very small chance that it could be helpful, but that's kind of like getting struck by lightning twice and winning the lottery in the same day. Now, because there are such incredibly large numbers of cells in the world, the numbers are actually pretty good. The statistics work out pretty well for there to be favorable mutations occurring at a fairly regular rate. But the vast majority of mutations are still going to be unfavorable. An incredibly small number or percentage of them will be good for the organism. As long as our environment is stable, our mutants are going to be only a very small percentage of the population. If there's a change in the environment, that mutant that had the mutation that just happened to give it an advantage to this new environment is going to be better able to survive in this new environment. For example, we can look at antibiotic resistance. If there are no antibiotics being used, the antibiotic-resistant bacteria are actually at a disadvantage. They are not able to reproduce as quickly as bacteria that are susceptible to antibiotics. But if you introduce antibiotics into the resistance, that's a new environmental trait that all of a sudden makes the environment favorable to antibiotic-resistant bacteria, and the non-mutated, non-resistant bacteria are now at the disadvantage. So one potential method we have for eliminating antibiotic resistance is to go 20-30 years as a human race without using any antibiotics. And that would make the environment favorable for the wild-type bacterium, and we could breed out the antibiotic resistance, but that's totally not a feasible solution. I can't think of anybody who would want to go 30 years right now without using antibiotics. That'd be very unethical to do. Let's have a concept check here. Class, which of the following types of mutations will have the most devastating effects on the cell? A point mutation, a frameshift mutation, missense, nonsense, or silent mutation? If you can't remember, and I'd be surprised if you don't remember this one right off the top of your head after watching this video, please rewind the video, check your notes, or check the PowerPoints, or check your textbook too. Alright, pause the video, find me an answer. 1, 2, 3, 4, 5. The answer is B, frameshift mutations. If you have any questions for me, please feel free to post them on the discussion boards or shoot me an email. Happy studies!